thanks very much for having me here. Good morning, good afternoon, and well, good night from Helsinki here in Finland. So glad to be here and to present to an illustrious audience tonight. I'm Philippe Arnez from CancerDiag, based in Finland, as said, and I'm here today to present to you Diagmmr, our novel method to detect Lynch syndrome. So I'll start with maybe talk about what Lynch syndrome is. Lynch syndrome is an inherited mismatch repair deficiency, and it's the #1 cause for inherited cancers. And it's also more dominantly inherited, so you have a 50% chance to inherit it if a parent has it. And people with Lynch syndrome have a very highly increased risk of developing cancer over their lifetime. The main cancers that are associated with Lynch syndrome are colon cancer and endometrial cancer, but the spectrum covers about 10 different types of cancers. So how is this diagnosed today? Typically, it's still the cancer which triggers the testing, and there's a whole bunch of tests that are done in normal practice, and then if it's indicated, ultimately they'll do genetic testing, and there's basically 3 outcomes that can result from that. The first one is the so-called pathogenic mutation is found, meaning that the person has Lynch syndrome. Unfortunately, there's many cases where they find either VUS or a so-called no mutation at all. And given that there has been reasons to believe that it could be Lynch syndrome, those are then suspected cases where no diagnosis is really possible. So why is that? You're all very familiar, I'm sure, with sequencing, but just to remember, we're trying to look at a very micro level in the DNA, in what these mutations that are found may affect at the end of the day in a protein. In the case of Lynch syndrome, these mismatch repair proteins, which is a very difficult feat to do. And with our new method, we take the proteins from the patient's cell and look directly at the functionality of those proteins. So no matter what has caused the problem in one of the previous steps, we will detect that. So we literally measure and quantify how well the proteins are doing what they're supposed to do. So this mismatch repair efficiency, how well that is. And if we measure that it's normal, then it's called proficient, so there's no Lynch syndrome. And if there's the deficiency, well, then it's deficient, meaning that the person has Lynch syndrome. So we directly assess that. And because our assay is based on a very small skin biopsy that we get from the healthy skin from a patient, we can do that for asymptomatic patients, basically meaning that we don't wait for a cancer to appear, but we can test people preventively. So with this, we really close an unmet need there in the current pathway. And a big opportunity arises also from preventive testing in the future, so that we can actually find those people at risk and test them before they develop cancer, so that we can prevent it. And so that is really where this end cancer moonshot comes in. What's the opportunity? The innovation has been quite slow in that area, and so it still costs a lot of money and lives. And the low diagnostic rates that we see that hasn't changed much. And it's estimated for the US, for example, there's about 1 million people with Lynch syndrome that are unaware of it, and this causes high morbidity, mortality, and of course a huge cost to society. The market that we have identified globally as an initial target market is about 500,000 tests a year, on which we can build on. Today, we have this method already launched in our domestic markets here in Finland as a lab-developed test, basically. So that is also a way we would see initial introduction to the US through a clear pathway. But for the future, we really want to scale and make it available in a decentralized way, and therefore we will develop a kit that is more scalable and more easy to distribute, and which will then require FDA approval. So that's all embedded in a big roadmap that we have. And currently we're actually raising funds to achieve all those big milestones that we've set ourselves. And this is what the money is going to be used for. So the method is validated already, it works. And what we now need to do is to show the utility really in the clinical setting. And we are launching a study here in Europe already this year. And so that's where most of the proceeds will go, but also into kit development and of course to back this all up a team growth. And we are looking at the US market. Obviously, we want to enter that too. And so we are looking for study partners in the US to do something similar that we're doing in Europe. And for the investment round, we are also open to investments from the US. To close up, that's our team. Just to highlight our founder, Minna Nyström. She's a professor of genetics and epigenetics at the University of Helsinki here, and has done more than 25 years of research in that area. It's very well connected with key opinion leaders in the field across the globe. So I hope that I was able to highlight the importance to find the people at risk that may have Lynch syndrome, and that our method can help to prevent cancer and to increase quality of life for those families.
Awesome. Thank you so much. Really amazing what you've accomplished. Preventative testing for cancer, that's a really transformative inflection point for cancer treatment. Logan has a question there in the chat. Logan, why don't you ask your question?
Thanks, Katya. Great presentation, Philippe. I was just curious, since the timelines can be extended in healthcare and in clinical research, what kind of timeframes are required for the clinical trials that you want to do and the approvals that you're working on? What kind of lead time are we looking at?
Yeah, so the study that we're starting in Europe, we kind of in the starting blocks. So we have a long history of collaboration with the university hospital here in Helsinki, so they are on board, but also we have a leading institution in Rome, in Italy, and with whom we have collaborated before, so some paperwork is done also there. We were lucky enough to identify a study in the UK, in Manchester, which was exactly looking at Lynch syndrome, and they have a pool of patients where we may access and provide that testing, so we don't need to start from scratch. So for this study here in Europe, we are in good shape, and we plan to get 300-400 samples this year from those locations. So we should be able to close that sometime mid next year, I would say. For the US, obviously it's a different story, because there we have to start from scratch almost, right? So, but we will collect a lot of experience here in Europe, so that will certainly also help.
And Philippe, can you tell us a little bit about the experience for the users in Finland? How has that rollout process been going, and what's the feedback you're getting?
Yeah, we have had a very good feedback on the test. There was one case. I had a call with the doctor before Christmas, who had ordered the test, and his case was exactly such as I illustrated, that they found a variant of uncertain significance, and they didn't know what to do about it, and so he ordered our test, and it came out negative. And so he explicitly said that it was very helpful, because he could discard that, and the patient had come in with the daughter, and they were really worried about what this means if she has such a hereditary condition. And so that was kind of a relief, and has well, helped the clinician obviously in the diagnosis, but also will hopefully improve the quality of life of the family, because they know that they do not have Lynch syndrome. And another case from Helsinki was that where a patient was diagnosed with cancer, and also again a VUS was found, and so we tested, and in this case came out positive. And retrospectively, after a couple of weeks, when talking to clinicians, we found out that we actually after that also had tested two daughters of this patient, and one had inherited the condition and the other not. So the daughters were young adults, so they did not have any cancer or anything. So we were able to kind of help that stratifying of the family members.